Hello everybody. Today we will be studying about the fourth type of hypersensitivity, that is the type 4 hypersensitivity. Now this is also called as cell mediated it is also called as cell mediated type of hypersensitivity, because here you can find that there are no antibodies involved in the entire process. Only your immunological cells are involved in the reactions. So that's why it's called as cell mediated. But whereas other types of hypersensitivity, you could find the involvement of some or the other type of antibodies, but here antibodies are not involved. Okay, now this is also called as delayed type of hypersensitivity, because it takes time for the reaction to happen. That is, within 1 day to 4 days it takes, not within few hours. But whereas the others, it takes within few hours the reaction is seen. Okay, now as I told, no antibodies involved, and the cells that are actually taking part here is T helper cells, T cytotoxic cells, macrophages, which act as APC also. APC is antigen presenting cell, and which also act as effector cells. So they have a dual role. Eosinophils are involved here. Okay, so these are the different types, these are the different characteristics of hypersensitivity type 4. Now in this type 4, it can be categorized into two main types. Okay, hypersensitive type 4 is of two types, that is cytotoxic type and macrophage type. From the name itself, I think you can understand cytotoxic type means the T cytotoxic cells are involved here, and macrophage type means macrophages are involved here. Now, what is cytotoxic type? Cytotoxic type means it results in contact dermatitis. Now, when the name itself you can make out, it is something related to the skin. Basically, it's skin allergy. Okay, now when we talk about this, which activates or initiates this type of reaction is your chemical substances. Only the chemical substances will result in this type of hypersensitivity. It could be soap, it could be jewelry, nickel, any kind of metals. Okay, only the chemicals, if they are there, this hypersensitivity will get activated. Now what kind of chemicals? Any kind. The chemicals should be absorbable type. It should get absorbed onto your skin, not ingested. Okay, now chemicals if ingested also your body reacts to it, but it doesn't result in hypersensitivity type 4. Okay, absorbable type. The chemical must be absorbable by what? By your skin, epidermal layer of your skin. Only then it results in cytotoxic type of hypersensitivity. Okay, now as a result of this entire process, what happens is the skin will start peeling out, it will start separating out, blister formation also will happen. Now let us see how exactly it happens. Now for that we have to slightly understand the different layers of the skin here. If you see the outermost layer of the skin that I have shown, it is called as a stratum. Okay, this all these layers are the epidermal layers. Okay, the outermost layer of the epidermis is the stratum. Little below to that is the granulosum, and these vertical cells are spinosum. Okay, stratum spinosum, and these the lowermost epidermal layer is called as bacillum. Now these are the cells, bacillum, these are the cells which are dividing type. Okay, now what happens here? There is a chemical, I told you, it results because of the chemicals. Okay, absorbed by your epidermal layer. So there is a chemical here I have shown in a dotted form. This chemical somehow gets inserted, it gets absorbed onto your epidermal layer, into your skin. Okay, and the bacillum, those cells take up the chemical on its surface. Okay, now this chemical is just a hapten. Now what do you mean by a hapten? Hapten is incomplete antigen. Okay, it cannot start your immunologic reactions until and unless it becomes an antigen. So it is not an antigen now, it is just an hapten. So what does this do is, it combines, that is this haptin, this chemical combines with another protein. There are many proteins on your skin, isn't it? It combines with another protein from your skin and then forms a complete antigen. Okay, now this antigen will now be presented onto the surface of the bacillum. Okay, now this bacillum now acts as an APC, antigen presenting cell. Okay, it now gets presented on through the bacillum with the help of the MHC class 1 molecule. Okay, now I think you understood what kind of antigen it is. I told you how it is made up of. Okay, now this is the skin cell which is presenting the antigen with the help of MHC. Now this complex now activates the T cytotoxic cells. Okay, it activates the T cytotoxic cell because the TC cells have receptors for the MHC class 1 holding the antigen. So as this complex is formed, now what happens is the TC cell, it itself is a cell which cytotoxic name itself tells. Okay, it when this complex is formed, the TC cells will now release out perforins. Okay, where it will release out into the surrounding. Definitely this perforins will go and attack the cell also, the APC. Now which is APC? APC is a skin cell now. Okay, now what is the action or activity of perforins? It produces pores, it produces holes. So these perforins will go and produce holes on the surface of the skin cells. Many holes are being produced. Now if many holes are produced, what happens to the cell? It will get lysed, isn't it? So that is what I have shown here. If you can see the lowermost, this is the place where the entire reaction was happening, isn't it? Now here you can see the all the cells are getting lysed. So this layer starts getting detached. Okay, so that's what I had told, dermatitis. The layer starts peeling off, it gets detached. Now because this layer starts getting detached, you can see here in the last diagram, all the other layers also, these layers also will start getting detached. A bleb is formed. Okay, and bleb is formed, that is a swelling, and then the layer, this skin layer starts getting detached. This all happens because these skin layers, these epidermal layers, the bacillum is, the cells are getting lysed. So that is the reason. So as a result, you ultimately will land up in the thing called as your contact dermatitis. Okay, the skin starts peeling off and blister formation. I told you how the blister also formation happens. Okay, now I told you the second type, that is a macrophage type. Now let us see when does this macrophage type gets activated. This macrophage type gets activated when there are pathogens involved. The first one was cytotoxic, was the chemicals involved. Here pathogens. What kind of pathogens? Not any, it has to be intracellular. Okay, it has to be inside your cells. That means Mycobacterium tuberculosis, Mycobacterium leprae, these are the bacteria which is present inside your cell, inside the host. They cannot be free living. Viruses, you know, they are obligate. They are always present inside the host. So these kind of pathogens will activate the macrophage type of hypersensitivity. Now let us see exactly what happens. There is a bacteria here. Obviously it enters into the host. Okay, now that has been engulfed by a macrophage. Now you remember your host means your body cell. Okay, it gets engulfed by the macrophage. What will macrophage do? It will present the antigen on its surface with the help of MHC class 2. Here MHC class 2 is involved. Okay, now this MHC class 2 will now activate the T helper cell. Okay, it will activate the T helper cell, not the TC cell. In the previous one you had seen that it will activate the TC cell, but here it activates the T helper cell. Now after the T helper cell gets activated, what happens is that your uh, a T helper cell will have receptors on its surface is which can bind to the MHC class 2 of your macrophage, carrying the macrophage. Okay, now this T helper cell will get activated and it will start releasing interleukin 2. Interleukin 2, when it gets released, in turn the macrophage will release interleukin. First one was the T helper cell releases interleukin 12. This interleukin 12 in turn will initiate the release of interleukin 2. Okay, now what this interleukin 2 will do, we are concerned about interleukin 2. It will help the T helper cell to proliferate, that means to increase in number. Who releases this? The macrophage releases. Where it goes, the interleukin 2, it goes to the T helper cell. Okay, now this T helper cell, when it gets attached or when it gets bound to the interleukin 2, it will help the T helper cell to proliferate. Okay, now this, not only to proliferate, it also releases, that is T helper cell also releases IFR, that is interferon gamma. What interferon gamma will do? It will activate the macrophage activity. Now what do you mean by macrophage activity? That is, it increases the production of nitric oxide and oxygen radicals. Usually the macrophages will kill the host cell or kill the antigens by production of nitric oxide. But here what happens, than the usual production or the normal production of nitric oxide, because of interferon gamma, the production of nitric oxygen or nitric oxide increases drastically. So this nitric oxide will drastically, because the number is increased, the quantity of nitric oxide increases, there is tissue damaged. Many tissues it will damage because it starts increasing. The nitric oxide production is more here. Okay, now this entire thing happens with T helper cell 1. Okay, now there can be another pathway that is followed in the macrophage type, that is this one. Now I have told you about this complex, isn't it? That's the macrophage which holds the antigen, acts as APC, the T helper cell gets activated. Now if T helper cell 1, TH cell 1 gets activated, this is the pathway followed. Suppose instead of T helper cell 1, the TH2 cell gets activated, then what happens is the T helper cell 2 releases out interleukin 4 and interleukin 5. There what was released out? The interleukin 12 was released out. Here, because T helper cell 2 is getting produced, activated, it releases interleukin 4 and interleukin 5. What interleukin 5 will do? It will activate eosinophils. Now you very well know what is activity of eosinophil, isn't it? Now what eosinophil will do? Eosinophil, the interleukin 5 will activate the eosinophil. Interleukin 4 will result in the degranulation of eosinophil. What is there in the granules of eosinophil? It gets released out. What is there? It has proteasomes, protease, histamines. You know that histamines, heparins, they are allergens basically, isn't it? They ultimately result in tissue damage. So these are the different types or pathways of hypersensitivity type 4. Thank you.